This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Today, we're talking about influenza and the controversy surrounding gain-of-function research.
This is, as you can imagine, a very controversial idea to study a virus and then try to make it more lethal.
This episode was recorded prior to This Week in Virology episode 294, where very similar issues were discussed from a slightly different perspective. If you're not a fan of TWIV, you probably should be, so go check out twiv.tv for more discussion. Hi, this is Audiommunity, episode 8. I'm Kevin Bonham, and joining me today is Kate Franz.
Hello.
And unfortunately, well, I guess fortunately for Matt is off doing job interviewee stuff because he's hopefully wrapping up his PhD fairly soon. He is not joining us, but today Kate and I are going to be talking about, well, I guess mostly Kate is going to be talking about influenza and a recent paper about trying to make really, really deadly influenza, which seems like a wonderful idea. So, Kate, why don't you take it away?
Yeah. So, I guess like Kevin said, we're going to be focusing a lot on influenza, a lot of virology today. So, I dropped in some immunology for you, Kevin.
Oh, thanks. I appreciate that.
But a lot of what we're talking about has to do with influenza pandemics. And so, when we think about influenza pandemics, you might think of 2009 and like swine flu. And was that so bad? Like, our pandemic's really a bad thing. What does a pandemic look like? What do we do to prepare for pandemics? And I think when virologists think of pandemics and think of like really bad infections, we often think about the 1918 influenza pandemic. So first, a pandemic basically means like a widespread infection. So it's not just, a couple people here and there getting infected. It's something that's going across the world. So in 1918, at the end of World War I, there emerged this strain of influenza that started infecting soldiers. And so it infected people in the United States, in Boston, in France, in Britain, in Spain, in Germany. So it was really widespread. Because there was a war going on, there was a censorship occurring, so no one could report that people were getting sick where there was fighting. But what journalists could report on was Spain, who was neutral during the war, and how all these Spanish people were dying of flu. So it became known as the Spanish flu, not because it really came from Spain or that it hit Spain particularly hard, but just it was the only place that anyone could talk about it.
Do we actually know what the origins were? Is it sort of similar to, like, modern pandemics usually arise in Southeast Asia and then sort of spread west and south from Southeast Asia. But do we have any sense of 1918? Are the records good enough that we can tell where it actually came from?
So no one knows for sure. There are a bunch of theories out there. Some people think that the 1918 flu came from France. Some people think it came from Kansas. Really what you look for is influenza is what we'd call a zoonosis. So it originates in an animal and then it transmits to humans or other animals. So the 1918 flu is thought to have originated in birds and then underwent some sort of evolution and then was able to infect humans. And in 2009, when we had the swine flu, that was a combination of birds and pigs, influenza strains, and then they were able to transmit to humans. So when we look for the beginnings, we look at where are humans interacting with animals that carry these viruses. And so Kansas was brought up because they thought maybe it arose on a farm, and similar for France. And like SARS was transmitted from humans interacting with fowl in marketplaces. So very, very close contacts where these viruses actually could find a human to infect.
Can we say something about the reason for that? Like, why is influenza always coming from an animal vector? I'm thinking sort of the evolutionary arms race sort of explanation. My understanding is that essentially you have influenza strains get into people, those people tend to start building up immunity, and so it sort of brings the levels of influenza infection down. Meanwhile, you have these animal reservoirs that are circulating and sort of recombining and reassorting different strains that can then jump out and get into humans who don't have any immunity to this new reassortant strain. Is that sort of right? Or
yeah, I think that's right. We constantly are having this exchange of viruses between humans and different sorts of animals. I mean, mostly like pigs and birds. But yeah.
But I guess what I'm asking, so we sort of think of it, the anthropocentric idea is that the animals are sort of breeding grounds for bad influenza that jumps into people and then it's in people. But is it more that we're just constantly sharing and we're infecting the animals?
So I was thinking about this because we usually do think of it as birds gave us the flu and then pigs gave us the flu, but are we giving any back? And apparently we do actually can transmit human strains back to swine and birds. I don't know how strong the interplay or how strong like that transition is. Like, is it mostly strains go into humans and then they die there, or do they pop back out and go back into the birds? So there's evidence that definitely happens. I don't actually have a really good feel for how common that is. Like, my guess would be that it's pretty common because we do see strains reemerge after like 30 years. So
right.
There are like very similar strains that come back and then and infect humans. So
okay, fair enough.
Okay.
Sorry to derail it.
Yeah, no, it's fine. That's like the point.
Right.
So 1918 influenza. So 1918 influenza infects 500 million people, and it was estimated that about 20 to 100 million people die from the infection. So the kind of conservative estimate is 50 million people. So 3 to 5% of the world's population dies from 1918 flu. This is one of like the greatest, not great as in good, but this is one of like the largest natural disasters in human history, is this pandemic of influenza virus. So with that history in mind, we spend a lot of time thinking about pandemics and how we can protect or respond if a virus similar to 1918 or a virus dissimilar but had a similar mortality or case fatality rate were to emerge.
So just as a comparison, the bubonic plague in the 14th century, over the course of about, I think, 30 years throughout Europe, the Black Death is thought to have killed about 30% of Europe's population, which was, there's literature, there's all kinds of things that are associated with this particular thing. And 30 to 60%, obviously that's a large number, but that's just of Europe. So total global population, the Black Death over the course of 30 years killed about 5% of the global population, 5 to 10% of the global population. Whereas in 1 year, 1918 killed between 3 and 5%. So this is not an insignificant thing. And it's kind of interesting to me that despite what a major thing this was, I think maybe because it was in the middle of a world war or something, but there's not as much sort of global memory around that event, as devastating as it was, as perhaps there should be.
Yeah, I agree with you. I think I definitely would have thought, oh, the Black Death, that is like our biggest infectious disease event that ever happened. But I think 1918 influenza gives it a pretty strong run for its money. I mean,
yeah, especially if you think timescale wise.
Yeah, especially on the timescale. So normal influenza, just seasonal flu, has a case fatality rate. And so case fatality means how many people who are suspected or confirmed to be sick. Usually this means like you have gone and seen a doctor. The case fatality rate is 0.1%, whereas the case fatality rate of the 1918 influenza is 20%. So huge increase in fatality. Like, this is a very virulent strain. If you looked at the distribution of death compared to the age, a normal flu is U-shaped. So there's high in mortality for the very youngest of the population, high mortality in the very elderly. But 1918 is actually a W shape. So there's some mortality in the young and the old, as there always will be, but there was a spike in young adults. So like the, 20 to 30 range. And so this was really strange because usually young adults have strong immune systems. They can fight off a flu. But in this case, what was thought to have happened, and this was tested when they resurrected the strain a couple years back in monkeys, and they think that the 1918 flu initiates some sort of cytokine storm. And so a very strong immune system makes a very strong response. And this is actually what was killing people, is their responses were too strong and it kind of ran away on them.
Yeah, this is the immunology portion of
this is the immunology for you.
Audiommunity.
Well,
yeah.
I mean,
so let's linger on that a bit,
yeah.
just because it's the it's the only bit that's going to be immunology. So this is sort of an interesting point. I think that normally we think about weakened immune systems being the bad thing in terms of infectious disease. Obviously, we know that allergies and autoimmunity are because of sort of overactive immune systems, but we don't generally think about heightened immune system responses being responsible for death in infectious disease. And I think this conclusion is a bit counterintuitive at first, that a strain that, because young adults have such strong immune systems, they were more susceptible to dying. One potential reason for this might be that when you die from influenza, you don't actually die from influenza itself. You don't die from the viral infection. Usually what happens is you get secondary pneumococcal infections. So you have bacteria that get into your lungs, and because of all the damage that the influenza virus has done, you are susceptible to bacterial infections, and it's the bacterial infections that kill you. So we don't normally think about this, but the immune system can cause a ton of damage.
Yes.
And normally for most influenza infections in young adults, normal circulating strains, the amount of damage caused by the virus is stronger than the amount of damage caused by the immune system. And because you have a stronger immune system, you can then also fight off the secondary bacterial infection. And this is simplifying a little bit. There's a lot of potential explanations for why bacteria can establish better infections during the course of influenza that we won't get into, I don't think. But it's sort of interesting to think that this influenza virus in 1918 was causing an immune response that was so strong that the immune system actually did a ton of damage to these people's lungs. And then that allowed probably the secondary bacterial infection to take over.
Yeah. I will say, I think something important to note with this sort of strain, though, is that the replication of 1918 happens in the upper respiratory tract. Of course, there is damage that occurs to your lungs. But I don't know if you remember, do you remember H5N1, the bird flu? That was this really interesting strain that replicated primarily in your lungs as opposed to your upper respiratory tract. So when people came in with confirmed H1N1 cases, they immediately started them on antivirals and antibiotics because they were at such extreme risk because there was so much more replication happening in the more sensitive part of their body. I thought that, because usually you hear like, you don't get antibiotics if you have the flu, but with H1N1, they're like, put them on antibiotics, put them on...
You mean H5N1?
H5N1. The numbers, we're going to have a fun time today.
Yeah, we should probably get into what that means exactly. But before that, just to clarify the point, we sort of normally think about the lungs are just one thing, but it turns out that there are cellular differences in the sort of upper parts of your lung and the lower parts of your lung. And actually, there are several different layers of different expression of surface things. And this H5N1 bird flu, the thing that it binds onto on the cells is a variant that's expressed more in the sort of lower deep lung, which is probably why it's not as infectious, and maybe you're going to get into that in a sec. But it also means that when you are infected, you are much more susceptible because viral infections that are happening deep in your lungs are more dangerous for various reasons than those that happen in your upper lung. So
we're talking a bit about the immunology of an influenza infection, but I want to focus a little bit on the virology. It'll also help when we discuss the paper to understand what the parts of the virus actually are. So influenza virus belongs to this group of viruses called orthomyxo. So ortho means correct and myxo means mucus, which is kind of interesting because that's mucosal surfaces is where this virus is found. The orthomyxoviridae family has 4 different groups. So there's influenza A, which is the one that's mostly causes influenza. It's the one that infects humans, causes the most disease. There's influenza B. There's sometimes some human infection with influenza B. And there's influenza C, which has like really no disease in humans. And then a tick-borne disease, the Goto virus, which basically no one knows anything about. So when people want to study orthomyxoviruses, they're like, let's study influenza A. Tick viruses are not interesting to us. So most of the research is done on the flu. And so
wait, are you saying that it's a tick-borne human infection, or it's a virus that infects ticks?
It is a virus that infects ticks. I'm not sure if it infects humans. I think it does.
Okay. Otherwise that'd be really cool if we could like soup up this virus and get it to kill off ticks that spread Lyme disease. But that's a different discussion. We can talk about that another time.
Yeah, so it's transmitted from ticks and it infects like guinea pigs. It can infect humans sometimes, and its genome is much smaller than normal flu. This virus, the way that we talk about them, so influenza viruses, they have 2 proteins on the outside of the virus. These are the envelope proteins, and this is actually how we designate the different strains of influenza virus. So there are 2 proteins found on the outside. One of them is called hemagglutinin, or HA, and hemagglutinin has about 16 different subtypes. So each different subtype has a number. So hemagglutinin 1 to 16. I think 2 more have been discovered recently, but we usually deal with 1 to 16. And then there's neuraminidase, and neuraminidase has fewer subtypes, and those are also distinguished by numbers. So when we talk about influenza H1N1, we are talking about hemagglutinin subtype 1, neuraminidase subtype 1. These subtypes are important because related subtypes seem to be controlled by the same immune response. So the 1918 flu was an H1N1. So the 1918 influenza pandemic was an H1N1 subtype. And in 2009, when there was another outbreak of H1N1, people who had been exposed to 1918 flu had some cross reaction. They had some sort of response to the 2009 strain.
Yeah, I just want to sort of linger on that point a little bit. So in previous episodes, we have talked about memory responses, and we're almost certainly going to be talking a lot more about them in the future. But this sort of is a wonderful demonstration that people who were, 90 years old, who had been exposed to 1918, I guess almost 2009. So people who were over 90 years old, who'd been exposed to the 1918 flu pandemic, still had lingering protection in their memory immune responses in 2009, which is pretty incredible, especially because we normally, like Kate said earlier, we normally associate a lot more death with people who are elderly. But obviously people who were really old and had been exposed to 1918 were sort of somewhat protected from the 2009
So I just want to say,
pretty amazing.
If you read Twilight, Edward died from 1918 flu. And so he was alive in 2009, theoretically, and he would have been protected if influenza could infect vampires. Just putting it out there. I can't talk 1918 flu and then say, oh, by the way, it wasn't the thing that killed the sexiest vampire that is known today.
Although
you can't
on that
cut
note,
this out, Kevin.
I don't know if you've been watching True Blood,
I don't.
But part of the whole thing that's happening in this current season of True Blood is hepatitis V, which is a engineered virus that infects both vampires and humans, but humans are asymptomatic.
Is this a new virus? Because I watched the first season of True Blood, and someone was like hepatitis. Did you say D or V? Okay, someone was hepatitis D positive, and it kills vampires. Like in the first season, this is like a plot point that someone went up to a vampire and was like, suck my blood, and they fed from them, and then the vampire died, and they're like, you're hepatitis D positive. Like the person just wanted to go kill vampires. Anyway,
that doesn't sound familiar to me.
Maybe I made that up.
Yeah. Anyway,
maybe that was my True Blood fan fiction.
or that was hardcore foreshadowing, because it's like the whole thing.
No, I think
this is way off topic. We can get into that.
Immunity After Dark.
Yeah,
where we discuss probable science in fantasy series.
But I have this section, so I'm teaching this infectious disease class at Emerson again this fall, and I want to have a section where I talk about like monsters, like werewolves and vampires, as infectious disease.
Zombies.
And I thought it would be like super meta. Yeah, zombies, absolutely. I thought it'd be super meta to then say, okay, and we're going to use viruses infecting vampires where the humans are asymptomatic as examples of like zoonotic transmission and things like that. So... But this is totally far afield, and we were talking about Edward being infected with 1918.
Yeah, we were talking about how he would be immune in 2009.
The fact that people who were infected...
I mean, maybe he wouldn't be immune because he did die from it.
Right.
But he probably died because he was young... He was like 18, or he was 17, because, the whole, like... interesting issues with the age differences, but he was like 17. He died probably from complications of like a cytokine storm. And then, he's now older. His immune system is theoretically weaker because he's 107 in the future. Mm-hmm. And so maybe he is protected. Maybe he doesn't die a second time.
But also immortal. So in any case, I just want to clarify cytokine storm. So we've mentioned cytokines before. Cytokines are the chemical messengers of the immune system. So when you have an infection, when you have inflammation, your immune cells are releasing cytokines, which is just a fancy term for these chemical messengers. They're sort of like the hormones of the immune system that are communicating between immune cells and saying, hey, inflammation is happening. And so a cytokine storm is basically overproduction of these cytokines. It causes things like vascular leakage. It causes swelling, inflammation, and a bunch of tissue damage, essentially. So if you have an overabundance of cytokines, it can cause serious problems.
Okay, back to the virology. So right. HA.
This is going to be real fun to edit, by the way.
Hemagglutinin, is one of the envelope proteins of influenza, and HA is responsible for binding the receptor of a cell that it's trying to infect and then mediating the entry. So HA actually binds to sugar residues, sialic acid, and the interesting thing is in birds, the HA binds sugars that are said to have a 2,3 linkage, and in humans, influenza HA will bind the sugars that have a 2-6 linkage. So 2-3, 2-6 linkage, what this basically means is these sugar molecules, if you were actually to draw them out, they kind of look like large hexagons. Is that the one that's 6?
Yeah.
They kind of, I don't know if it's a if it's a hexagon or a pentagon, the aromatic acid.
But they're rings. They're ring structure. Sialic acid
so they're
ring-shaped
rings.
sugars.
And so the different carbons that make up, so the different atoms that make up the rings, we just number them 1, 2, 3, 4, 5, 6, 7, 8, 9. And so 2-3 linkage means that the second atom of one of the rings has a bond with the third atom of another one. And so this is just a way to identify like what the chain looks like.
And essentially the take-home message here is that they're just different chemical shapes.
Yeah, exactly.
Right?
I like
so normally we think about binding, proteins binding to other things is a shape sort of interaction. And there's chemical interactions, there's bonding affinities and stuff like that. But you can sort of think of it like lock and key. And the key that is the receptor should bind into the lock that is the ligand. And there's some shape affinity. There's some, not all locks fit all keys, obviously. But the point here, this 3-5 versus, was it
2-3 versus 2-6.
Or 2-3 versus 2-5, 2-6. So these different bondings is just basically that a single key is able to fit two different types of locks. It's like a skeleton key a little bit. And it's different in birds and humans.
Yeah, so so there has to be some sort of change to the HA protein in birds before it can infect a human. So if the HA protein comes from a bird, it needs to switch so it can now fit the shape of the 2-6 linkage, and then it will be able to infect a human cell. Oh,
I see. So it's not that it's the same key that's fitting two locks. It's that there's two different locks. So in order to use you have to like saw off of one of the teeth of the key to make it fit. My analogy earlier, I said it was a skeleton key.
Yeah, no, it's not a skeleton.
You're saying
key.
it has to change.
Key. No, so this is this HA. So this HA is important because, like I say, it determines which sugar it can bind, and when we switch from animals to humans or birds to humans, it needs to also switch in order to infect properly. And so we say HA is now the species tropism determining factor. So whatever your HA is going to determine if you can infect a human or a mouse, or a human or a bird. I'm so used to the human mouse comparison. But a human or a bird.
Yeah, no, for sure. Right. And we mentioned tropism before in the context of viruses infecting different cells in the body. So you can say in a person, some virus may have a tropism for neurons versus lung cells and stuff. But we also use the term tropism to say different species. And so bird tropism versus human tropism. And within those species, they then also have a cell tropism.
Yes. So all your human glutenins do the same thing. They all are going to bind sialic acid. But the different HAs are going to look slightly different from one another. They're going to their immune system is going to respond slightly different to the different subtypes. So H1 and H5 do the same thing, but they look slightly different. And I think those are the most important things to understand about HA. The other protein that is on the surface of an influenza virion is called neuraminidase, and it cuts the sialic acids off of HA when the virus is actually leaving the cell. So if the virus is leaving the cell and it's covered in sialic acid, it's not going to have HA free to bind to a sialic acid residue of a new cell. So this is just like an
Would the viruses get stuck on the surface of the cell that they just left as well? Yeah. Or like try to re-enter?
They would get stuck. Or they would just be covered in sialic acid and now they can't infect anything. But yeah, it helps the virus's bud to have active neuraminidase. So the other thing that neuraminidase seems to be able to do in some viruses, and this makes them super virulent, is if neuraminidase can help the HA molecule become active, not by just removing sialic acid, but actually cutting it so it now becomes able to enter into a new cell. Usually that's done by the protease called plasmin, but that might be a little too high detail. If you're hanging with this, good for you.
Though I want to ask, do you think that this is sort of an off-topic question, but you mentioned that there are less versions of neuraminidase than there are of HA that we're aware of. Do you think that's because neuraminidase is an enzyme, it has to carry out a chemical reaction. Do you think that it's just more constrained in the sorts of shapes it can take because it has to have this enzymatic activity, so it's a little less susceptible to changing shape than HA?
No. I mean, you could hypothesize that might be true. It's not necessarily true. Neuraminidase is a tetramer, and so you have 4 proteins bound together, and the active site is on the top of the tetramer. So it basically looks like 4 boxes pushed together, and so the surface top of the box is the active site where all the chemistry happens.
So all the sides could maybe be changed.
So you could think that there might be some sides that could be changed. And I mean, HA is also constrained in that like it has to retain its binding to salicylic acid. I think where the difference is happening is that HA, so a concept in virology is that viral evolution is driven by its interaction with the immune system. So a virus is not going to evolve unless something is stopping it from doing what it wants to do. So HA is like this very, comes in contact with the immune system much more frequently than any other viral protein. So HA has to like go and bind to a cell, and then that lets the virus get into the cell and start to replicate. So HA is a really great target for the immune system to stop the actual entry of the virus. So I think it might have to do more with like the importance of HA in the viral life cycle.
Right, the selective pressure
there's more pressure on HA. So that could drive a higher evolution. But it might be, I don't know too much about what the active site actually looks like for neuraminidase, and it might be that active site is highly constrained for whatever reason. I mean, so neuraminidase cleaves sialic acid. So like they are binding the same thing. So the binding pockets could be similar.
Right. Yeah, but I guess that's a good point. If antibodies were constantly being directed against neuraminidase, we'd almost certainly see. I mean, we do make antibodies against neuraminidase.
We do see, but
it may not be as detrimental to the virus if you're binding to the neuraminidase.
Yeah, I agree. So other proteins that make up an influenza virion, so you have the outside of the virus, you have neuraminidase, you have hemagglutinin. Then on the inside, and then there's a membrane that makes up the virion. Then on the inside, you have these 8 genome segments. So that's the genome of the virus. Those are the instructions on how to make itself. And that encodes 11 proteins. And these proteins are mostly just proteins that direct the replication of the genome to make more virus. And so I guess, so of particular interest for today, some of those proteins are called PB2, PB1, and PA. And those are the polymerases. So they are in charge of binding to RNA and then making an RNA copy of that RNA, which can then be used to translate protein. So it's the nucleic acid code that then directs the protein construction. And then...
So can I just interrupt real quick? So that means that it's a negative strand virus?
It is a negative strand virus.
Okay. So a confession, I have heard virologists say negative strand versus positive strand RNA viruses for ages. And until the last episode where you and Matt were talking about... LCMV, and you described the difference between positive and negative strand virus. That was the first time I had ever heard an explanation of what that actually meant. So just to restate, even though you explained it wonderfully in the last episode, negative strand means that you have to first make an RNA copy of the genome, which is then the template for protein.
A negative strand virus has all of its information for making the protein in the reverse orientation that you want it. So you have to make an exact mirror image of the actual RNA strand in order to get it in the right orientation to make your protein. So if you were to translate a negative strand piece of RNA, you would just get nonsense. If you could even translate it, if it was even if it even had like the correct signal to start translating. So influenza, they take the negative strand, they first make copies of the negative strand to give it a positive strand orientation.
So it's essentially like making mRNA.
It's like making it looks like host mRNA.
So just to get a little bit more into detail on the mechanism here. So in human cells, we have DNA as the template, and then you make the complement of that DNA strand. So DNA has this base pairing thing. RNA has the same base pairing thing. And so in mammal or in most things other than viruses, the genome is encoded in DNA. And then to make protein, you make a sort of copy of the DNA that's going to be a mirror image of the DNA code. And that is the template that is then used to make the protein. And so negative strand RNA virus seems like it's sort of the same thing, except that it's RNA instead of DNA that's the template. Whereas a positive strand virus, it can just come out of the capsid and immediately start making protein?
Yeah,
is that the
deal? It actually, it's interesting because there are these themes that kind of, there are these themes in the viral life cycle that break down by, are you a positive strand or are you negative strand virus? And it has a lot to do with the way that the genes are actually oriented. So if like certain genes are encoded at very beginning of a strand of RNA, RNA or the very end of a strand of RNA or DNA. So that's why people actually even talk about negative or positive stranded viruses, because you know that they're going to have certain life cycle features. But that's a lot. That's like super basic virology. I mean, that could be its own like little figure for the site.
Sure. That's not a bad idea, actually. But so I was just wondering, like, why wouldn't all viruses just be positive strand? Because it seems like so much easier. But it occurred to me that if you're a positive strand virus, you still need to make the negative strand to then be a template for the genome, right?
One, viruses just don't always do what's easiest. But one reason why you might have a advantage to be a negative stranded RNA virus is, and I wrote about this and in commentary, PubMed my name, it's up, is that
actually why don't we include that link in the show notes just so people can.
In a positive stranded virus, the first step is translation. And so when a virus enters a cell, you're only going to have like 2-3 genomes at most. Mostly you're going to have one virus genome in one cell. And one of the first things that the innate immune system will try to do when it senses a virus has entered is shut down translation. So if you have one genome and translation has been impaired, your chance of actually getting a full translated variant goes way down. But if you are a negative stranded virus, you're sensed as you enter the cell. Your first step is not going to use the translation machinery. Your first step is now to replicate all of your RNA, and you're going to make a ton of RNA. In fact, you're going to turn off cellular mRNA production, and you're going to just route everything and make viral mRNAs. And so now when the translation machinery is turned off, you have thousands and thousands of mRNAs, and your chance of getting just a small percentage of those translated goes way up. And if you have enough mRNAs, you might be able to override a translational block, if that makes sense.
Right, that's a really good point. So and we should say that when you say translation is turned off, what you mean is it's turned way down.
Yeah.
And so
I would say it's reprogrammed. I think that would be the safest way to say it. It's not off, because obviously there are still things that are happening, but it doesn't run canonically. So certain proteins.
Right, but it's not like a light switch. Yeah. So it's not just gone. And so if you're using that negative strand template to just make crap tons of message copies, whereas if you're a single positive strand entering a cell, then you have one thing to translate proteins off. Whereas if you've made a bunch of copies off of your negative strand, you've got maybe hundreds or thousands of copies that have a chance to get copied. That's a super interesting idea. I hadn't thought about it. But sorry to derail the conversation again. But it's nice. We're adding more immunology into our immunology podcast about viruses, which is
okay. So
they would never talk about this.
No, they wouldn't. Okay, so we have the components of the virus that are important. HA enters a cell. There are proteins that replicate the RNA, and then they make the messages that will become the proteins. They also make new genomes that will be bound by protein and then placed into new variants, so the whole cycle can start all over again. Okay, so what I want to do next is just talk about the paper and a little bit of context surrounding this paper. So the paper is called Circulating Avian Influenza Viruses Closely Related to the 1918 Virus Have Pandemic Potential. And this is coming from Kawaoka's group in Wisconsin, University of Wisconsin-Madison.
We should say the first author too is Tokiko Watanabe. Yes. Or Watanabe.
So it was about 10-15 years ago that a study was published, and in it they resurrected the 1918 sequence of the virus from a female victim of the 1918 pandemic who was frozen in the permafrost in Alaska. And so they found this woman, and they were able to sequence out the virus genome. So we said that there are 8 segments, and they found the sequence to all 8 segments. And I think that this was an interesting study in that they made the virus, and now they had the sequence, and there are systems and techniques that allow you to take the sequence of the virus and actually make the whole virus. It's really simple.
So would it be, so we've already talked about vampires in this episode. Maybe we should have waited for Halloween for this. But would it be safe to say that this is now a zombie virus?
It might be a zombie virus. Yeah, they resurrected this virus. Though
so it's not that the virus causes you to be a zombie, it's that the virus itself
the virus was basically Han Solo'd. Yeah.
Nice.
Oh yeah.
Nerdtacular.
So yeah, they found someone who was a victim who had died and was frozen, and then they were able to resurrect this virus sequence. So now we know what the 1918 virus looks like. Studies have been done where they infect monkeys with the 1918 virus. It's where they confirmed that this virus actually does cause a very powerful cytokine storm. And in 2009, we had another pandemic of the swine flu, and this was also an H1N1 virus, like the 1918 virus. And I think this really sparked some new interest in studying the 1918 virus. Or at least, I think it sparked a lot of awareness about research that was happening with these viruses. And so because we had this pandemic in 2009, it's very similar to 1918. This is very scary because 1918 was an awful disaster. We have a lot more attention now onto how can we prevent pandemics. And so a lot of groups think that a good way to prevent pandemics is to study the viruses that have pandemic potential, like H1N1.
It's sort of interesting. I feel like for a while before swine flu happened, it was like H5N1 all the time. People were like, H5N1, bird flu. Like, oh, bird flu is really scary. And then all of a sudden H1N1 swine flu happened and everybody's like, oh crap. Oh, it turns out that it doesn't necessarily have to be a bird flu for it to be scary.
Yeah. Okay, so I mean, people are interested in potential pandemics, and so H5N1 was like this very, very high mortality. It seemed to replicate, like I said, like low in the lungs, which is a very dangerous place for it to replicate. But because it replicated so low in the lungs, it didn't seem to transmit from person to person very well. You had to get really close to someone or like very close to an animal source of this virus. I mean, to transmit person to person, you like literally have to like cough up a lung.
And just taking this back to what we were talking about earlier, so is this, I may be wrong here, but this is when we were talking about the locks and keys and how birds have different locks than humans do, it's not that humans have different locks, it's that in birds, the lock that this H5N1 could bind to is in the upper respiratory tract of the bird. So it can transmit easily from birds, but that same lock, that same sialic acid thing is in the low lungs in humans, not in the upper lung. Is that right?
Yes.
And we're going to get into using ferrets for experimentation, but the reason that ferrets were used, or the reason that ferrets are often used in studies of influenza is because the distribution of their sialic acid in their airway is very similar to humans. Yes,
that is correct. Because H5N1 has like this interesting attachment, it replicates low in the lungs, it wasn't a very good pandemic virus. It didn't transmit very well. But if H5N1 were to be able to transmit well and still maintain its virulence, this would be a very scary virus to have loose. And so some people think that it's worthwhile to study H5N1 and see if this is even a possibility for it to become more transmissible while keeping its lethality. This is, as you can imagine, a very controversial idea to study a virus and then try to make it more lethal. And so these sorts of experiments are called gain-of-function experiments, where you take H5N1 and you mutate it and you try to make it more transmissible. The way so there are two groups that did this. One of the groups was Kawaoka, whose paper we're going over today, and the other group was Ron Foucher's group in Erasmus Medical Center. And these papers, when they were reviewed and were to be published, they actually were held up for publication for people to actually discuss whether or not we should be releasing information that could make these viruses potentially more hazardous to humans.
Yeah, this was like a huge controversy back in summer of 2012. Yes. There were these two papers that did very similar things, and they were basically held up by Nature and was it Science? There were two separate papers, one in Nature and one in Science. These are basically top tier journals, and these journals were asked to not publish the specific sequences, and it was this huge controversy. A lot of scientists were saying like this is scientific censorship, and a lot of people were saying, yeah, but like, do you really want to be giving people the ability to make these really deadly viruses? As Kate mentioned, you can go from the sequence of a virus to making the actual virus pretty easily. I mean, it's not trivial, but it's not like
pretty easy if
impossible.
You are trained. It's very easy if you're trained.
Yeah,
so
certainly.
I mean, you do need to have specialized training because
you need specialized equipment and you need specialized training, but you don't need. It's not like a nuclear reactor. Like, you don't need super specialized parts. You couldn't make it in your garage, but if you have a molecular biology lab and the right training, you could do it. I'm not arguing that it was the right thing to do. I don't think it was. And actually, back in 2012, a graduate student group that I'm in called Science in the News, we did a presentation, and I'll put links to the videos to that in the show notes so people can take a look to sort of get more about that controversy, or just Google H5N1 research around 2012 in the summer. And or actually, I think it might have happened in the winter. And then we talked about it in the summer. But anyway,
there are a lot of interesting concerns
it was
over
a big deal.
The bioethics of making a virus that is more deadly, the safety concerns. And should we actually be making viruses that have dual use? Like maybe they give us some sort of benefit, but at the end of the day, does the benefit of researching these viruses really outweigh the risk of either someone intentionally or unintentionally releasing the viruses out into the public? I don't know. My thoughts on this whole controversy was like, if I were to make a bioweapon, I would not make influenza. Like, I would not use influenza. There are so many better bioweapons available. Like,
yeah, but that's not an unreasonable. That's not an unreasonable point.
I mean, yeah,
right?
Okay, let's go real talk / we're gonna get censored by the NSA. Okay, if I were to make a bioweapon,
hi guys.
Okay, so I could take influenza and I could lyophilize it, right? And I could release it into the air. But people are gonna inhale it and then exhale it. So really, one of the big issues of making a bioweapon is making something that will attach into the lungs when you inhale it. So it's this very complicated physics problem. A problem that I will say the US government has really nailed. So
yeah, no, that was one of the things when I was teaching this infectious disease course. I went and looked at all of the examples of governments using bioweapons. Apparently, I forget if it was Japan or China that attacked either Japan or China. I forget which directionality it was, but basically they flew planes and like dropped bacillus spores, just spread it over the countryside from planes. Like, it's sort of, people have used bioweapons, and it's sort of,
the history of bioweapons is not that great in terms of it being very successful.
Lobbing plague bodies over the walls of forts.
Yeah,
which apparently there was this one particular battle that probably sparked the 14th century Black Death pandemic in Europe, because the invading army lobbed plague bodies into this city that they were sieging. People escaped from the city out to sea into the Mediterranean, or actually out into the Black Sea, I think, and then eventually into the Mediterranean. And that's sort of where the Black Death spread from. So
that's great.
Anyway.
So, out of this controversy came the U.S. Department of Health and Human Services made these guidelines on what sort of research should you publish that has a potential to be dual use, meaning it could be helpful, it could be harmful. And so they suggest that research should be funded if the virus anticipated to be generated could be produced through natural evolutionary process. The research addresses a scientific question with high significance to public health. Third, there are no feasible alternative methods to address the same scientific question in a manner that poses less risk than does the proposed approach. 4, biosafety risks to laboratory workers and the public can be sufficiently mitigated and managed. That's an interesting one. 5, biosecurity risks can be sufficiently mitigated and managed. 6, the research information is anticipated to be broadly shared in order to realize its potential benefits to global health. And 7, the research will be supported through funding mechanisms that facilitate appropriate oversight of the conduct and communication of the research.
Yeah, and a lot of that is fairly straightforward. I mean, that those seem like common sense guidelines. The one about that it could plausibly happen through natural evolution, that's a little strange, because there are a lot of cases in which we would want to make changes that aren't likely to evolve, but are still nonetheless very, very useful in terms of their research potential. So that one I'm a little bit shaky on, but the vast majority of those seem perfectly reasonable to me.
I really like the natural selection one, and I think those 7 guidelines are, like, keep them in your head when you go through the paper and see if you think that this paper meets those guidelines. Obviously, the government thought that this paper met those guidelines because it was published, but there have been some, like, bioethicists, and in particular, there's a Harvard professor, Dr. Mark Lipschitz, and a Yale professor of epidemiology, Alison Galvani, and they have been very outspoken in saying that the studies that Ron Fouché and Kawaoka have undertaken do have realistic alternatives, and that they don't actually meet these dual use guidelines for research. And they very recently wrote a journal article just enumerating their different points of what are the alternatives to these experiments that we're seeing. There definitely is and
then Yoshihiro Kawaoka basically gave them the bird and published this paper.
Yeah, basically. Okay, so let's actually get into the paper because
Yeah,
I
let's
have
do
to
that.
go to work at some point today. John isn't here. It's a
Yeah, I should
boss
say
free day today.
Nice. Yeah, I should say that we're recording this in the morning, which is unusual. Normally we record in the evening, and which is why last episode we talked all about what we were drinking. Probably doesn't make sense to start drinking at 10 a.m. even when we're recording Audiommunity. So, Kate, what are you drinking this
Drinking
morning?
coffee with 1% milk because I'm out of half and half.
Oh, nice. All right, very good.
And what are you drinking today?
I am drinking also coffee, but this is a coffee called Jet Fuel that we have little Keurig makers downstairs at work. So it's called Jet Fuel, and it's fairly appropriate. It's thick and black and super high caffeine, which is wonderful. I was making the joke to someone earlier that science is basically fueled on drug use,
absolutely,
and it's legal drugs. It's caffeine and, well, the ones that we talk about are caffeine and alcohol. Those are my main two vices, but it's sort of interesting that it's perfectly acceptable to talk about how much caffeine you drink and how overcaffeinated you are, but it would not be equally appropriate to, for instance, talk about all the Ritalin you're taking or, all the amphetamines.
I would like to say, for the record,
it's essentially the same thing. It's performance-enhancing
crackers.
drugs.
I'm a big carb eater. So those are my drugs of choice. Pizza is also
Nice.
always accepted. I actually
Agreed.
have had coffee and pizza as a meal multiple times. It's really weird. It's a weird combo, but I like both of them.
That sounds wonderful,
I like
though.
both of them so much. Together, they're not great, but separate, man, they're so good. But like, I've definitely had times where I'm like, I need caffeine and I need carbs, pizza and coffee.
I want to say for the audio listeners, while Kate was saying how much she likes caffeine or coffee and pizza, she was doing like a motion, like shoveling things into her face.
That is true.
As they were doing it. Okay, so the paper.
Yes, the paper. Okay, so just to repeat it, the paper's title is Circulating Avian Influenza Viruses Closely Related to the 1918 Virus Have Pandemic Potential. So it's a very strong title, I think. Basically saying
And
1918
straightforward.
can happen again.
Right.
And so I would just like to start with the first sentence of this paper. I highly recommend just trying to read the introduction of this paper. It sounds like it was written by Bill Pullman in Independence Day. So it starts with, despite having conquered many infectious diseases, we continue to face a threat from novel, previously unrecognized infectious disease. This is great. Like, it starts there and then it just takes off. So, yeah, someone should be a speechwriter for the government.
Yeah.
Okay. But in this...
I mean, he's throwing down the gauntlet. I mean, there's all this controversy. I can sort of understand why they wrote it like this, because there's been all this controversy about this dual use stuff, and they basically want to say, like, no, you bastards, this needs to be done.
Yeah, and well, I think we can go through the paper and we can see if they adequately address concerns. So one of the main idea behind this paper is that they hypothesize that because 1918 was this awful pandemic, that if there were sequences of 1918 viruses circulating in the avian population, in the bird population, they could theoretically reassort and make a new virus that could be transmitted to humans that would be 1918-like. And if this happened, would our current vaccines and therapies be able to combat this virus? And so
Can I just talk real quickly about assortment, because you didn't get into it earlier.
I was going to, and I kind of skipped it.
So Kate mentioned that these viruses are segmented viruses, which mean they have multiple different, sort of like chromosomes. They have different genetic segments. So rather than their whole genome being on a single strand of RNA, there are multiple strands of RNA that are packaged into a single virion, and they're sort of like chromosomes. There's different RNAs that encode different proteins. And so one of the things that can happen with influenza is not only do you get sort of normal mutations through different replication cycles that cause some what's called drift in the types of proteins that are in influenza, you also have the case where if a single strain, or if two different strains of influenza infect the same animal, and in particular they infect the same cell, when you get replication of the different RNA segments, you can get a new virion out that has some segments from one virus and some segments from the other virus, sort of like viral sex in some senses. So you could have, for instance, if you're infected with an H1N1 and an H2N3, you could end up with an H1N2 N3, and also all of the other proteins. The H and the N are the ones that we sort of talk about often, but you can also end up with different polymerases, different other accessory proteins.
Yeah, as long as they're encoded on separate segments. So hemagglutinin and neuraminidase are on separate segments. Some segments have 2 proteins, so those proteins wouldn't reassort, but everything that's on its own segment will be able to reassort and make these interesting combinations. So there's this probability that if there are 1918-like segments circulating, even if they are in separate viruses, eventually they could recombine and we could remake that same virus again. And now I would say that depending on what the actual prevalence of each of these sequences are, there's going to be a different likelihood that could happen. So if the 1918 sequences are only found in like a percentage of a percentage of a percentage of birds, this would be a very unlikely scenario. But if certain segments are found in like 5%, 10%, 20% of the birds, then it becomes much more likely. Okay, so the first thing that they do in this paper is they BLAST, which is a basic local alignment search tool, which is basically like a very complicated Google search for nucleotide sequence, or they look at protein sequence, not nucleotide. But they BLAST the 1918 protein sequence against all the different flu sequences that we have. And they found that many of the proteins from 1918 that they blasted, so this is I want to make this point. So the 1918 virus is a human virus. It's adapted to humans, and they're blasting it against the bird virus sequence. So when they say 1918-like, it's like the bird flavor of the human virus. So these still are bird viruses. We're not talking about a human virus that's ready to go. So this would still need a significant amount of evolution before it could infect a human.
Yeah, and I just want to say real quick to emphasize this point. So we have this giant database of essentially all of the DNA sequences or RNA sequences of all the different viruses, of all the different animals, of all the different plants that have been sequenced in sort of global research. People deposit the sequences that they get into these giant databases, and then we can use computational tools like BLAST to then say, okay, like how many other genes or other genomes are there like this one that I have? And I think just want to highlight that because biologists, this is sort of like standard, but I think it's really cool. And it's something that I don't know a lot of people are aware that we have these just probably now billions, if not hundreds of billions of nucleotide sequences. I meant nucleotides that have been sequenced, not individual sequences, but we have a lot of information in databases that we can look at computationally. And that's kind of cool because it allows us to do things like this.
Yeah, it's a great tool. I mean, I think every biologist uses these sorts of searches, at least once a month.
Yeah.
Okay, so they search the 1918 sequence against this database of bird influenza sequences. And so they see that many of the proteins of the 1918 virus have a high homology, meaning that they're still highly similar to a protein that is currently circulating within the birds. And interestingly, a lot of the proteins were actually recently isolated in 1995, 2002. These aren't sequences from the 1920s that were there in the birds when the first jump happened. These are sequences that have been maintained in the population and they're still currently circulating. And so there are 11 proteins in influenza. And so just kind of like a quick rundown. So the polymerase proteins, they had 8 changes in one protein, 6 changes in another, 20 changes in another, and 9 changes. Some of these proteins called the non-structural proteins, which are responsible for combating the immune system, they had 5 changes, 4 changes, or one protein had no changes. So they were able to find proteins that were very similar to 1918. Interestingly, their HA and neuraminidase protein, so the H and the N, they had 33 and 31 changes, which is a lot. And it's not that surprising that HA and NA would be so different from a 1918 strain because these proteins are undergoing the most evolution. These, like we said earlier, they're coming in contact with the immune system, they're evolving fast, they're going to make a lot of changes in order to still be able to be maintained without being cleared. So it's unsurprising, but I think it is important to note that the HA, which is this determinant of tropism, is very, very different from the 1918 virus.
Yeah, this is one of my criticisms of this paper, at least the way it's presented. And maybe this knowledge is common knowledge in influenza research. I don't know. But they talk about how closely related these avian influenzas are to 1918 because they're only, 7 amino acids different. But they don't talk about a normal circulating H1N1, like compared to swine flu. How different is swine flu in these proteins in terms of their amino acid homology? Because, if normal circulating strains have like 20 changes and these strains only have 7, like that seems kind of interesting. But if normal circulating strains have 9 changes or 6, then 7 isn't actually that interesting. And they don't actually address that. They might do it in the supplements. And Kate and I were talking about before we got on, the supplement for this paper is like 100 pages long.
It's really big.
A nightmare to drive through. So they may have explained it somewhere in the supplement, but I think that's an important point.
Yeah, there is like some phylogeny that's in the supplement and also in the paper. But I think your point with comparing it to the 2009 virus is interesting because they do bring up the 2009 virus. 2009 swine flu is also H1N1. So how close is this virus to another human H1N1? Does this virus look the 1918 like bird virus? Does it look as similar to other H1N1s as it does to the 1918? I think that's really interesting.
Yeah, and this information is out there. Like
you could just blast it yourself.
If I took the time, I could blast it myself. But
I'm not.
When you're publishing a paper, I think it's important to include those caveats because they make a huge deal. They say 1918 like.
Yeah.
1918 like over and over and over again. These things are 1918 like.
Yeah.
Could I call swine flu 1918 like by their definition? Like, what's their definition of similarity? It just rubs me the wrong way that they wouldn't make that pretty obvious comparison.
Yeah, I agree. I think that would have been interesting information to have. That like what does this virus look like compared to other things as well? Yeah. I mean, I guess the one thing is that something like you'd have to compare it to other bird strains, and when it does, and we'll talk about it in just a second, when they do use a control normal wild type bird strain influenza, like a naturally occurring one, but they don't address like the sequence similarity in the main text. Okay, so they have this collection of genes that are most similar to 1918 human virus, and they decide using like these very simple techniques to make this into an actual virus in cell culture. And so they take the segments and they are able to put them in cells and get this 1918 like virus out. And so the first thing that you really want to do now is see, does this virus behave like the 1918 virus?
Before you do that, real quick, so they've picked these different segments from all these different things. Did they explain how they made the choice of this segment from this virus? Was it just essentially the versions of these proteins that were closest to 1918 that they combined those? Yeah. Okay. So each segment they blasted, they found the most similar to 1918 that was in circulation. Yeah. And they combined those. Yeah. Okay.
So what they do is they take the 1918 like virus, the 1918 human virus, and then an H1N1 naturally occurring avian influenza virus, which is found in a duck, and it was isolated in Alberta. So it's shortened to DK/ALB. So I'll probably just call it DK virus throughout this. And they take these viruses, they make stocks of them, and then they put them into a mouse. And they test at what dose of virus do 50% of the mice die. So they have like a group of mice.
Is it mice or ferrets at
mice.
This stage? Oh, it is mice. Okay.
They take groups of mice and they infect these mice with differing doses of virus, increasing doses of virus, and then they look for the group in which, at what dose did 50% of the mice die? And so this is called an LD50, so lethal dose 50%. For the 1918-like virus, they found 50% of the mice died at a dose of 300,000 virus particles, or PFU, which is plaque forming unit. And then it's
basically live viruses.
Yeah, basically viruses that are alive. So they needed 300,000 viruses to be put into a mouse before 50% of them to die. For the DK virus, they needed 6.3 million virus particles. So they need a lot of virus particles in order to kill 50% of the mice.
300,000 versus 6.5 million. Yeah. So it's basically 20 times more, right?
Yeah. And then for the 1918 human virus, they need 500 particles.
Whoa.
Yeah. So what we could say here is the 1918 strain is much more lethal than a naturally occurring avian strain, and it is significantly more lethal than the 1918-like strain, though there is a significant amount of lethality with the 1918-like virus. So in the paper they say the 1918-like virus has like an intermediate pathogenicity.
Which sort of makes sense. I mean, this is well, it certainly makes sense that the 1918-like virus, so these are segments combined from a bunch of bird circulating viruses. It makes sense that those would not be as lethal as the real 1918 flu that came from a mammal that we know was super deadly. That part is sort of unsurprising. It is interesting that if you combine the right segments together, you can get a 20-fold increase in lethality, at least in mice.
Yeah, it's interesting. I mean, it definitely says that there's something to this virus. Like this virus, it does tell us that what we were talking about before, okay, like how different is the 1918-like segments really, like, how different is it from other bird flus? And this data would say, though we don't know, like we weren't given the data on like the sequence, at least like in the pathogenicity, it is significantly different. There definitely is something here going on with the pathogenicity.
Yeah, the actual, we should say the actual numbers aren't super relevant because, this is mice and this is this weird hybrid virus, but it's just the concept that you have a significant increase in lethality with these particular versions of the proteins.
Okay, so the next model that they investigate is the ferret model. And so the ferret model is an interesting one because ferrets have a similar distribution of that's sialic acid that we were talking about in their lungs. It's similar to humans, and they also have similar symptoms. Well, I think the correct term would be signs of disease as humans.
Right.
Ferret symptoms are things that you complain about, and signs are things that you can see. And obviously, ferrets are not complaining about anything, so they can't talk.
Not in ways that we understand.
Not in a way that we understand. Yeah. And so they take the 3 viruses and they infect the ferrets, and they measure how much weight they lose. And so this is a really common metric to assess the severity of a disease in an animal model is to track what their weight is over the course of the infection. And so in the 1918 virus, the ferrets lose on average 17.5% of their weight. It's pretty significant. If you lost 10% of your weight, you would look quite different. The 1918
God, I want to lose 10% of my body weight.
Do it slowly. Do it slowly.
Right,
Don't get the flu.
Not through infection.
Yeah. Don't infect yourself with flu.
It's hard to keep those pounds off when you lose them because of flu.
The 1918-like avian strain induced a 12% weight loss, which is, very similar to the 1918-like loss. But the DK strain only induced a 2% weight loss, but that is plus or minus 3%. So that's essentially no weight loss. So there's no real disease with this natural bird flu. Additionally, when they inoculated these ferrets, they inoculate 3 ferrets in each of these groups, and one of those ferrets died from the infection. But the 1918-like avian and DK virus, no ferrets died. It's 3 ferrets. It's hard to say that that's significant, like statistically significant, but it is interesting. Also, these aren't done in duplicate. This is just like they had 3 ferrets, they infected them, and one of them died.
Right. Yeah, and as Matt said, sometimes animals die in experiments. For reasons that are unrelated to your experimental protocol.
It might be.
So it's hard to say 1 out of 3.
Yeah, and it might be if you do this experiment again, none of them die. Maybe one of them dies in 1918. This is like the value of repeating experiments. You don't do it in this case. They didn't do it in this case because they are using an expensive animal model, and I think it's fair enough to limit the amount of animals that you're going to sacrifice on.
Yeah, and the weight loss differences are significant enough. I agree. You're going to get to this later, but there's another strain that they generate that has giant weight loss, over 20%, and none of those ferrets died. So I think the weight loss percentage, it's a little more quantitative, and you can get a little bit better sense of how virulent these things are. The death is interesting, like you said, but it's not super important.
Yeah. So again, they say that the 1918-like virus has an intermediate pathogenicity. It seems to create a disease better than a normal bird flu, but it's not quite as bad as the 1918 human virus. And so they hypothesized that they could increase the pathogenicity of the 1918-like virus by substituting in the different 1918 segments into the virus. So essentially what they're saying is there are 8 gene segments if we could take one of them and replace them with 1918, if that increased the weight loss of these ferrets, we would say that segment is responsible for the increased pathogenicity of 1918 over 1918-like. So they make these viruses where they replace the bird polymerase with the 1918 polymerase, they replace the bird HA with the 1918 HA, and then they do a couple combinations of all of these. And they see that when they add the polymerase gene, PB2, they increase the weight loss. So it goes from 12% to 17.8%, which is very similar to the 1918 loss. And then when they add the HA from the 1918 strain, they see an increase again of weight loss to 17%. They also see an increase of mortality.
Yeah, although the variation goes up too. So when you just have the polymerase, it's 18% with a 2%, like plus or minus 2%.
Yeah.
When you add the polymerase and the HA, it's essentially 19% plus or minus 4%. So you get more variation it's a slight increase in weight loss. I don't know how significant
I think I meant increase compared to 1918 like avian influenza.
Right. Oh, yeah. Fair enough. Yeah.
But you're right. The HA alone, that might not be significant if you were to do this a couple more times, because the variation is really high with the HA alone. But you do see increased mortality. So 2 ferrets die.
Yeah, true.
When they have the flu that has all of the 1918 like bird flu segments, but has a 1918 human HA. From this, they conclude that PB2 and HA can increase the pathogenicity of the 1918 like avian influenza. The next sort of concept that they go about investigating is transmission. So transmission is key in a pandemic. H5N1 was not a pandemic because it didn't transmit very well. They want to look at these viruses and say, are any of these viruses good at transmission?
Put it in sort of starker contrast. You think about Ebola, a virus that everybody is freaked out about because it's so deadly and so terrible. Ebola has killed less people in 40 years that we've known about Ebola than influenza. Like by several orders of magnitude, it has killed less in 40 years than influenza kills in a single year. Like a normal run-of-the-mill yearly influenza strain kills more people than Ebola has in 40 years. And that's because influenza normally, even though it's not particularly virulent, it is so transmissible and infects so many people that a lot of people die from it just because it spreads so well. So
the actual number on it is about 35,000 people a year will die of influenza if it's just a normal seasonal influenza strain.
Yeah, and that's in the US alone, I think. I think it's hundreds of thousands of people around the world.
Okay.
Yeah.
Okay. So they want to look at the possibility of these viruses being transmissible. So they have this kind of cool setup. The way that they look at transmission is really interesting. They take a ferret and they shoot 1 million virus particles up its nose. It's the same way they did it in the first part of the study. And they wait a day, and then the next day they put a ferret in a cage next to the infected ferret, and they're far enough away that they can't directly touch each other, but they are within coughing distance of one another. And so what you do is you track, and they call this ferret, the uninfected ferret, the contact ferret, and you track the virus titer in the contact ferret over a set of days, like 9 days, and you see if that contact ferret becomes infected with influenza. When they infect a ferret with 1918 virus and then they put their contact ferret next to it, they see that 2 out of the 3 contact ferrets become positive for virus. 2 out of the 3 contact ferrets also create antibodies against the strain of influenza. When they do the same experiment with the 1918 like avian flu, none of the contact ferrets become sick. When you do a normal avian flu, the DK flu, none of the ferrets actually get sick. And then again, they look at their reassorted 1918 plus 1918 like avian flu. So it's like a little bit human, a little bit bird flu, the ones that we talked about earlier. And again, if you just add the PB2, the polymerase, or the HA, none of the contact ferrets become infected. So they try now, and this is really the controversial part of the study, is they now take this 1918 like avian flu and they mutate either PB2 or HA. And they do these mutations that have in past studies shown to increase transmission. They also increase tropism. So they basically take the avian flu, the 1918 like flu, and they make it look more like the 1918 flu. So they make it look more human. And if you mutate certain amino acids in these proteins, they see that you can increase the transmission. So if you make a mutation to the PB2 or 3 mutations to HA, you increase your contact ferrets transmission to one ferret becomes infected. 1 ferret transmits. And if you make a couple more mutations to your HA, 2 ferrets get virus transmitted to them. They become infected.
So this seems super relevant because as opposed to a 1918 virus segment all of a sudden appearing in the ferret, having the segments that already exist and then making a few mutations to the hemagglutinin, which we know happens all the time. I mean, hemagglutinin is fairly malleable in evolutionary terms. That seems fairly relevant. It seems like something that could happen in nature if you get the right reassortment where you get this 1918 like flu. Changing the HA by a couple of amino acids is not out of the realm of possibility.
No, and in fact, they think that the 1918 flu that was transmitted to humans actually was a circulating bird strain that had one small mutation that made it more likely to infect humans. And that was the way that it was transmitted. It was transmitted through a small mutation as opposed to other flus that are the segments all reassort and now it's more likely to infect a human. This was 1918 originally was a flu that was thought circulating in birds, not causing any problems. And then 1 or 2 mutations happen and now it's really great in humans. Like it replicates really well in humans and now it's transmissible. So the idea that this could happen, I don't think is all that unlikely because there are so few mutations that need to occur. These are also mutations that have been seen to occur before. And these PB2 mutations, the polymerase mutations, this mutation is circulating in the avian flu currently. So
I mean, we can go through this. We can go through the rest as well. But it seems to me that this is basically the punchline of the paper. I mean, they go through and they characterize the infections. They talk about where the viruses are. They're in the trachea or the lung. They talk about some of the pathology, like what is actually being damaged. But this is basically the take home. Am I wrong?
So I think this is what everyone took home. But they do go on to figure out what these mutations are doing. Like, why does this mutation actually make influenza more transmissible? They do say that. And so they show that this polymerase mutation makes influenza replication much more efficient, and that when these mutations in the HA make it allow it to bind the sialic acids that are found in humans much more readily. So they don't just make these viruses and say, boom, they transmit. Then they go on to like actually do the science, which I think is important, and show what are they doing? Why is this important? And then after they show what the mutations do, they say, are these mutations existent in nature? And they are. And so there are caveats to that. I don't want to just say like there are now 1918 is happening again. There's some caveats to that.
It's coming. That's a really great point. And
I think why I find myself leaning more towards publishing this sort of research is I do think that the science is good. Whether or not there are ethical alternatives, I think is a bigger question. It's a different question. But if you eliminate the controversy of dual use, this is good science. This isn't just like crappy science, and obviously we shouldn't do it. I think the controversy comes because this is interesting science. This is good science. We are learning something from it. We are learning about what makes these viruses transmissible, but there are these other scientists and bioethicists who think that this information could be learned in a different way. So I think one way to do this would be to take 1918 and try to cripple it. Take the human virus and now try to make it not transmissible. Are you able to make a virus that loses its transmissibility and then try to show specifically that virus, whatever you mutated, whatever you changed, that's the determinant of the transmission. Now that's harder. So the problem with that is like it's harder now to figure out what exactly confers transmissibility, because now I could break any virus. If I delete the polymerase, the virus isn't going to transmit. It's also not going to replicate. Right. So that isn't good information. But if I take a virus and I give it the ability to transmit, now we know exactly, or we have at least one component that we know is important. So it's...
Yeah, and in this paper in particular, if you don't make a virus that is more infectious than 1918, we know 1918, the sequence is out there.
Yeah.
So if I make a version of influenza that is more virulent than current circulating strains, but not as virulent virulent as 1918. Is that worse? Does that somehow make things worse? Because if you are a bioterrorist that wants to make a deadly agent, you're going to go for the most infectious thing out there, right?
Yeah.
Most deadly thing out there. So if I make these intermediate lethality viruses, I can then learn a bunch about what causes that increased lethality, what causes the increased transmissibility, but I haven't actually increased the danger more than if I was just working on 1918.
So I think the problem with that is that you don't know what you're going to make until you test it. So you could hypothesize that this won't be as infectious or as deadly as 1918, but you don't know until you actually test it.
Okay.
I mean, in these mutants, the mutant viruses that they make, they do see, out of the 3, they kill 2 ferrets instead of 1. So there's like this increased mortality. So maybe you argue that these viruses are more lethal than 1918. I think another really important point is that this is in ferrets and not humans.
Right.
And ferrets are not humans. And we don't know exactly how closely a ferret will mimic the human infection.
Yeah, we can certainly say that ferrets are among the best animal models, but they're still animal models.
Yeah.
So
and it's still only 3
That's a really
ferrets.
good point. Right.
Can we just look at 3 ferrets and say, well, this virus killed 1 more ferret than this virus out of 3? Obviously, is that strong enough? Is that a strong enough measure for your overall mortality?
Yeah.
I don't know.
On Twiv, they're always saying mice lie and monkeys exaggerate, and I feel like they came up with something for ferrets too when they talked about I
remember
on Twiv,
that. My thought is like a ferret is a ferret. I think they say like ferrets are not humans or something like that.
Right. Yeah, we will definitely link to those episodes as well because there was some good discussion back in 2012 when those original controversial papers came out.
Yeah, I think they actually talk about the supposed 1977 pandemic that maybe was leaked from a lab, which is what all these bioethicists are talking about. I never knew that was really a thing. I thought it was a frozen sample, but there's this whole controversy that maybe a virus has leaked from a lab before. But I would say that is a high maybe.
Yeah, right.
Maybe. Yeah.
And it's, even if it's true, even if we knew for certain it was true, it's still a sort of risk-benefit thing. I mean, biosafety in 2014 is much better than it was in 1970s.
Biosafety was still a developing concept in the 70s. You know, it wasn't really until recombinant DNA came out that people started thinking, oh, we need to have like measures in place to make sure something isn't going to fly out into the normal world. Yeah. I think
mouth pipetting was still happening in 1970s.
Mouth pipetting still happens. I have mouth pipetted. But biosafety, these bioethicists that I referenced earlier in the podcast, they cite examples in China that there have been leaks of virus out into the community, and that there are a couple cases where a researcher infected themselves and then they infected someone else, and that biosafety itself is not enough to regulate. That this is like, it doesn't stop anything. If everything is working up to the standards, okay, that's great, but there's always the case of human error where it doesn't work anymore.
Cool. So no, I was going to ask if there's more you wanted to say on the paper.
I did want to say one other thing that I thought was important, was that not only do they look and understand what these mutations are doing, they then take these mutant viruses and they see, are they sensitive to our best drug that we have against influenza, the oseltamivir or Tamiflu, and are they sensitive to serum that comes from H1N1 vaccinated patients from 2009. And so the way that you do testing the vaccine efficacy is you take the virus and you incubate it in sera from people who received the 2009 vaccination against H1N1. And then they take these viruses and they put them onto red blood cells. And so if the red blood cells clump together, so the virus is binding the different sialic acids on the red blood cells and they clump together, that's called heme agglutination. It's like the aggregation of heme, which is blood. They usually use turkey blood cells because turkey blood cells have the 2,6 and the 2,3 linkages, so they work for all influenza. And so if the virus that's been incubated with the serum can no longer aggregate the red blood cells, we say that virus has been neutralized, and that's usually by the antibodies that are in someone's sera. And so they show that all of their 1918 viruses and their mutants do have some sensitivity to the H1N1 vaccine from 2009, which is interesting. It's not as potent as when you look at the actual virus that the vaccine was directed against, but you do have some protection. So I think that they answer like two important questions with their mutants, which is like, would a vaccine work and would a drug work? They at least are being responsible in finding information that's important for public health knowledge.
Yeah, like you said earlier, they're not terrible scientists.
Yeah.
They're doing
Yeah.
they're doing good stuff. Whether it's stuff that is worth the risk, that I will leave to bioethicists. I tend to think that more science is better, and the risk is not zero. There is risk, but there's risk associated with a lot of types of biomedical research, and I don't think that the risk of this stuff is so much greater that it justifies not doing the research. At least not doing the research responsibly.
Yeah, I tend to agree with you. I read through some of the ethical alternatives to this sort of research. My personal feeling is that they do not match the sort of data and sort of information that you can get from actually these gain of function. But it's hard because it seems to be a matter of opinion. Like, there isn't a really good way to go head to head and say, I was able to figure out this information by doing this more ethical experiment. A lot of the ethical alternatives are really like, we should just not focus on studying these viruses, studying potentially pandemic viruses. A lot of it's like, we should just focus on making a universal flu vaccine. Then it doesn't matter anymore.
Right. Yeah.
I wonder how you test your universal flu vaccine without looking at potentially pandemic viruses, but that is an alternative that's proposed.
Yeah, but it's basically impossible to say, like, in 5 years, which research is going to be the most important for human health, right?
Yeah.
Like, you wouldn't have said in 1910, you wouldn't have said that research into fungi and how they're interacting with bacteria would lead to the greatest advance in human health in human history, antibiotics.
Yeah.
You wouldn't have said that studying a virus that infects bacteria would lead to all of this knowledge about DNA and the ability to do recombinant DNA.
Genes.
And, so it's
like,
I mean, it's impossible to say now what is going to be important 10 years from now in terms of research that's being done. But yeah, it seems to me, again, it's a risk benefit thing, and I think the risk is not so much higher that it is not justified.
It's definitely something that bioethicists are probably more equipped to deal with than myself. But at the same time, how many times do you consult with a bioethicist before you do an experiment? Zero. So it really is on the researchers to be thinking about these things and seeking out opinions to like learn more, try to figure out if there are more ethical experiments to be doing.
Yeah, at the very least, we should be having the conversation, because it is not reasonable for us to say that the ethics don't matter. I'm just saying, like, it's one thing for scientists to argue one side of the ethics, and it's another for scientists to be dismissive. And we are not advocating that scientists be dismissive of the ethical concerns.
Yeah.
But yeah, cool.
Okie doke. I don't know. Do you have any last thoughts?
I don't.
Okay.
And this has been over 2 hours of recording.
Yes.
So. And Kate needs to get into lab.
Yes.
So with that, this has been Audiommunity episode 8. I'm Kevin Bonham with Kate Franz. Matt will be joining us next time when he is back from his interviews at all different places in the country. We are on iTunes. You can search for us, Audiommunity. If you didn't know that, I don't know how you found us in the first place, but we are also on Facebook. I'm on Google Plus and Twitter. Kate, have you been doing anything with the Facebook page, or is that just one of those perennial?
No, I haven't done anything. I guess I just don't know what to do with it, which is that's like basically my problem.
I don't know. I mean, we're, like Matt said in the last outro, we're a little bit social media delinquent at this point, but that's okay. We are located in those places. Whether or not you'll find any value there is a totally different question. But when Matt and I do make graphics, which hasn't happened for a while, all the links to all of our social media and all the graphics and all the podcasts can be found at emunity.org. That's EMMUNITY.org. I should also mention we've asked you to send in email questions and comments, which we would love to receive, and you can email us at our first names at emunity.org. So Kate, Kevin, or Matt at emunity.org, and please do that and let us know if you have any questions or comments. We will happily answer them by email or talk about them on the podcast the next time. So we are supported in part by Harvard Medical School, and the music at the beginning and end of the podcast is composed by Rachel Reineck. We'll see you next time.